Okay then, this is just a short video to finish off the specific immune responses by lymphocytes. So in the previous lesson on Friday, we talked about the role of the B lymphocytes in the production of antibodies. We're going to move on now to T lymphocytes. So I think we're finished off on this slide on Friday. And this shows that full screen. This shows the B cells being activated when the antigen, you can see antigen number 1 there is binding to number 2. This is the antibody on the surface of a B cell. That stimulates the B cell to undergo cloning, so there's a really rapid cell division by mitosis to produce lots of plasma cells and a few memory B cells. Those plasma cells are like differentiated B cells. They've got more of the components necessary for protein synthesis, so they have more rough endoplasmic reticulum with lots of ribosomes and Golgi apparatus and mitochondria, and all of those things are useful for producing and packaging antibodies and then releasing antibodies for which can go on to neutralize viruses and bacteria, stop them from reproducing or to stop them from binding to a host cell. They can be useful for agglutination and precipitation, so sticking bacteria together for agglutination, sticking soluble particles together in precipitation. And then the memory B cells, which hang around in the blood or in the lymph for years and decades afterwards, so that if this antigen comes back, it can be recognised more quickly to produce a secondary response. Right, the reason I'm showing you this slide is one of the other things that can activate a B cell, so stimulate a B cell to undergo this cloning by mitosis, is one of the T cells. So this is an activated helper T lymphocyte, or helper T cell, or T helper cell. Okay, let's skip through that. So we're going to look at how those T cells then can become activated, what the 3 different types of T cells are, and what the function of each of those T cells is, and how they can assist in the in the response to a pathogen. Okay, so we mentioned this in the previous lesson. All lymphocytes are produced in the bone marrow. The B lymphocytes mature in the bone marrow, that's why they're called B lymphocytes. The T lymphocytes move from the bone marrow through the lymphatic system to the thymus, and it's in the thymus where they mature. So T lymphocytes, that T comes from the thymus. Okay, so here's just another little slide showing that. Okay, so we've also seen this slide before. This is about how the antigens can be recognised. So on a B lymphocyte, on the surface of the lymphocyte there are antibodies. As we said last time, those antibodies have those variable regions which are specific to just one type of antigen. And they're complementary to that antigen, and if an antigen binds to the antibody on a B lymphocyte, it stimulates the cell to undergo cloning by mitosis. T lymphocytes are a little different. They don't have antibodies. Instead, they have a different type of protein, a protein receptor. And again, those protein receptors are really variable. There are millions of different proteins, all with different shapes. that will be complementary to just one specific antigen. But like we said last week, there may be very few T cells in the body that actually that possess that specific receptor. So, on first exposure to a new antigen, say from a new virus, it may take some time before that lymphocyte is located, before the T lymphocyte is located, or the T lymphocyte comes into contact with that antigen. And therefore, those symptoms that you might experience when you first pick up a new pathogen may last for a few days, which will be enough time for that T lymphocyte to be found. Okay, so T cell activation, similar to the B cell activation, they can be activated directly by an antigen. Okay, so an antigen bumps into a T lymphocyte, but you can probably imagine that is fairly unlikely. What's more likely is that they are presented by that product of phagocytosis. So we can see here, this is an antigen presenting cell. Because as antigen presenting cells move into the lymph, move towards the lymph nodes where those T lymphocytes might be found, and they move between all of the different lymphocytes, presenting the antigens, hoping to find the correct one. When they do, the T lymphocyte is activated, and very quickly after activation, the T lymphocyte or the T cell divides again by mitosis. Now, on your assignment, you must describe that the B cells and the T cells divide and are cloned by mitosis. This is important because if they divide by mitosis, they are identical. In the B cells, that means they produce identical antibodies, all complementary to the same antigen. In the T cells, they have the same identical protein receptors on their surface, again, complementary to the same antigen. Okay, so as they're dividing by mitosis, they can differentiate into different types of T cell, T lymphocyte. There are cytotoxic T cells, or what are sometimes referred to as killer T cells. You can imagine what they do, but we'll look at that in a bit more detail in a minute. There are memory T cells, and the memory T cells, just like the memory B cells, they remain in the blood and in the lymph for months and years after that infection. And then there are the helper T cells, and we've already seen the helper T cells and their function. Their role is to stimulate and activate the B cells and stimulate those B cells to undergo mitosis, to divide, to produce plasma cells, which then go on to produce antibodies. Okay, so let's start with the cytotoxic T cells. These cytotoxic T cells are most effective against cells which have been infected by a virus. So as we know, viruses require a host cell to replicate. So viruses don't possess any ribosomes or Golgi apparatus, any of the machinery necessary for protein synthesis. So what they do is take over a host cell and use the host cell's cellular machinery to produce new viruses. The cytotoxic T cell is able to destroy and neutralize those infected cells. So when a cell is infected by a virus, more often than not, that infected cell will present a few of the antigens on its cell surface. An activated cytotoxic T cell will move through the body looking for these infected cells, and when it comes into contact with that foreign antibody that is complementary to its receptor on the surface, that leads to a series of events that eventually causes the cell to lyse. So inside the cytotoxic T cells are molecules called perforins. Now these are proteins which are released from the cytotoxic T cell and directed towards the infected cell. So that's a host cell, a cell from the person that's been infected by this new virus. The perforins land on the cell surface and cause the surface of the infected cell to become leaky. They do what's called lysis, okay, or they lyse the infected cell. And as they do that, the contents of that infected cell spill out, and that prevents that infected cell from doing the protein synthesis necessary for making new viruses. So it's like burning down the factory. This is stopping the infected host cell from making new viruses, and therefore slowing or preventing the spread of new viruses in that individual, but also hopefully preventing the spread of viruses to other individuals. Okay. As well as perforin, there is also an enzyme which is released, which is called granzyme. And granzyme works sort of with the perforin. So if we imagine the perforin is making the cell really leaky, the granzyme is responsible for what's called apoptosis, which is programmed cell death. Now all cells have genes which, when they are expressed, when they're turned on, produce proteins which kill that cell. And it's a really important feature of all cells. It means that cells don't age, or when they stop performing their functions properly, they undergo cell death, and that stops them from doing anything that might potentially be harmful. What the granzyme is able to do is switch on those genes, bring about apoptosis in the infected host cells. Okay, and there's a little slide here showing how that happens. Now, I'm not going to go into this in lots of detail, but feel free for your assignment to look up the role of granzyme, and you could give a short description of the function of that granzyme. Okay, so the second type of T cell is the memory T cell. It says in this first sentence, they're produced in the primary response. So primary response just means on first exposure to a new pathogen with new antigens, or antigens that haven't been recognised before, some of the cells that are produced by mitosis are memory cells, and then they remain in the blood and in the lymph. So instead of having just perhaps one T cell with the corresponding receptor on its surface for this new virus or new bacterium, there might be tens or hundreds, thousands perhaps, of these memory T cells that remain in the blood. So when you come to evaluate your evaluate the specific and non-specific immune responses, you can talk about the benefit of having memory cells, because it means that the secondary response, so the response that comes about after being exposed to an antigen for a second or third time, is much quicker, and that's because there are more memory cells than there might have been the original B cell or T cell. Okay, so here's a quick summary of the role of the T cells. We've not mentioned this third one yet, not in too much detail anyway. So an antigen presenting cell may present its antigen to a T cell, causing that T cell to undergo cloning by mitosis. It produces cytotoxic T-cells, as we mentioned. So they kill the infected cells by making those holes in their cell surface membranes. And the granzyme which is able to bring about programmed cell death. The memory T-cells, which circulate in the blood and the tissue fluid, ready for a future infection. And the helper T-cells, which stimulate the B cells to divide. Okay. Those helper B cells play a really important role, because without those helper T cells, the response by the B cells might be much slower. And it might mean that there is no response from the B cells. And if the B cells are not presented with the antigen, or they're not stimulated by the helper T cells, they might not go on to produce antibodies. And without those antibodies, the infection lasts much longer or can be much more serious. It's much more likely to cause damage to tissues or even death, I suppose. I'll skip through that and that and that. Here's just one last little bit then about the role of the helper T cell. So when a helper T cell or a T cell is produced, and those helper T cells are produced from mitosis, they produce these chemical signals called interleukins, or interleukin 2. And that has a few roles. Firstly, it stimulates the T cells to undergo cell division by mitosis, so they produce more and more and more T cells. It also activates the B cells, stimulating those B cells to go on to produce plasma cells by mitosis, and then those plasma cells produce lots of antibodies. And thirdly, they stimulate the cytotoxic T-cells, so they increase the number of the cytotoxic T-cells and stimulate them to go hunting for infected host cells to destroy them. Okay, I'm just going to skip through that. These two slides, they are on the PowerPoint. They're about these new. This first one's about a new type of cancer therapy called CAR T-cell cancer therapy, which is really interesting but not really relevant to this part of our course. And these are different types of antibody, which I did mention briefly in the previous lesson. I will just mention this. One reason why viruses, or you can be infected by similar viruses at least, over and over again, like the flu, is they have a really high mutation rate. And that means if their RNA in the flu virus mutates, that changes the shape of these protein spikes. Those protein spikes are the antigens, so they're no longer recognized by the same B cells and T cells that might have recognized them with a previous infection. And that means each new strain of flu virus appears like a completely new disease, which is why you might get flu over and over again. And also, it's why there are lots of different strains of coronavirus, the SARS-CoV-2 virus, which is why we continue to see these peaks and troughs and new types of infections and new strains of the coronavirus. Now, I'm going to talk a little bit more about immunity and infections for Friday's lesson, or perhaps the following Monday's lesson. Some diseases we may only get once, like chickenpox or measles, and that's because they have quite a slow or very slow mutation rate. So the virus doesn't produce any new antigens. So the memory cells, when they become activated, they quickly divide. They produce B cells really quickly and plasma cells, which produce and secrete antibodies really quickly. And therefore, that secondary infection, never has chance to do much damage. Some diseases that you do get more than once, colds and flu, coronavirus, is because of those mutations. They produce different antigens. And because they produce different antigens, the memory cells don't exist. That means that the primary response is required, and this takes longer. Now, the reason I wanted to talk about this is because for your evaluation, you need to, one of the drawbacks, I suppose, of the specific immune response is that it does take a while before there are a sufficient number of B plasma cells and cytotoxic T-cells and T helper cells to fight off an infection. Whereas the non-specific immune responses, so the barriers and the phagocytosis, they happen immediately. I suppose on the flip side of that, the non-specific immune responses cannot remove a virus or bacteria alone. They can only just suppress it and try to slow down the rate of infection. Whereas a specific immune response with the production of antibodies and with those cytotoxic T-cells, they can completely neutralise a new disease. Okay, this is what I mean by the primary and secondary response. This is a measure on the y-axis here of the antibody titer. That means the concentration of antibodies in the blood. So, with the first exposure to a new antigen, there will be no antibodies. Over a period of time, those B cells will be located. They start to divide by mitosis. They produce the plasma cells. And once there are those plasma cells there, they're secreting about 2,000 antibodies a second. Okay, but they only survive for quite a short period of time. But what we do notice is with the secondary exposure, we've got a slightly higher concentration of those antibodies. And the reason for that is because those memory cells remain in the blood for weeks and months and decades. And we can see those memory cells, when they become activated, we get a much bigger response. So a lot more of the plasma cells are produced, partly because you're starting from a larger number in the first place, but also because the secondary response is much more rapid. And the antibody titer, the antibody concentration, tends to remain higher for longer periods of time. Now, if we think about the vaccination program for COVID, we're all advised to get our first and second and then booster vaccinations. And that's applying this principle. So with the first exposure, you will have some immunity and you will have some memory cells that remain in the blood after your first vaccination. With the second vaccination, the number of antibodies that remain in the blood might be even higher. Actually, with the current COVID vaccines, the antibodies have declined a little bit, and the way that the SARS-CoV-2 virus has mutated has meant that it's been necessary to get a third booster shot, and that's the case with some viruses. So if you remember, just before Christmas 21, we're all advised to get a third booster shot. I think some over 75s now are currently having their fourth, aren't they? Okay, so I'll skip through this. And that's that. Okay, so that'll do. Okay, so on Tuesday's lesson, I'd like you to finish off the PowerPoints and your presentations for non-specific responses and the two specific responses, so B cells or the humoral response, T cells, or sometimes called the cell-mediated response. And then in Friday's lesson, I'll send you some of these details. I'd like you to move on to the distinction work, which is evaluating those responses. I'll put that all into a rubric. I know one or two of you asked me to do the rubric, but I've just not had a chance to over the weekend, but I will make sure it's there for Friday's lesson. Okay, take care. I'll see you on Tuesday the 21st. Cheers, bye-bye.